Well, good morning, everyone, to Grand Rounds this morning. A few housekeeping items. If you'd like to receive CME credit for your attendance today, the access code is 166351, and this can be found in the chat feature at the bottom of your screen. For questions, please send Katiadu Fofana an email or drop a note via the chat feature. Today, it's a great pleasure to have Dr. Heath Skinner from the University of Pittsburgh Hillman Cancer Center join us for Grand Rounds this morning. Dr. Skinner is a leader in head and neck cancer treatment and a leader in radiation biology, which is a topic that's, I'm sure, dear to many of you. He completed his MD and PhD program at West Virginia University and subsequently moved to MD Anderson, spent quite a bit of time there and established himself as a researcher with funding, significant funding from the NIH. He has about 8 total grants funded thus far, focused on lung cancer and some head and neck cancer, and then moved very recently, I was very surprised to hear that, has been at UPMC since 2018 only, and is now the chair of the Department of Radiation Oncology. Heath, I don't know how he can find time for his work. He is very busy. He told me yesterday he has about 60 faculty he needs to evaluate. On top of that, he has an active lab, 2 active R01s that he is working on. I don't want to extend this introduction too much, but I would say we were very happy to have him here and happy to hear his talk. And thank you, Heath, for coming all the way to Atlanta.
Well, thank you very much for that very generous introduction. Honestly, as I mentioned at dinner last night, I think the busiest part is the 4 soon-to-be 5 children. So I think that actually occupies the majority of the time. But thank you very much for everyone here and everyone online. I'm going to be talking to you today about histone acetyltransferases, how we might be able to target them, and how they affect responses to DNA-targeted therapy. And largely focused on radiation oncology because I am a radiation oncologist. And, when all you have is a hammer, everything looks like a nail. But let me begin my preface with just discussing a little bit about some of the challenges in head and neck cancer. Unlike many other malignancies, we are a bit hamstrung in regards to what we can molecularly target. We all know that HPV positivity is associated with an excellent outcome, but heretofore, and again, there's many trials on the horizon looking at circulating tumor HPV and a bunch of other stuff related to HPV, but right now, it's not really impacting a lot about what we're doing in the clinic. And then with HPV negative malignancies, it's even worse in the setting of the vast majority of these mutations that we see in tumors or mutations in tumor suppressor genes, which are extraordinarily and notoriously difficult to target. Moreover, the utilization of genomically driven selection or thinking about things in a genomic perspective is also proved challenging. Just as an example, we know p53 mutations, the great-great-great-great-grandfather of genomic alterations in solid tumors, present about 80% of HPV negative head and neck cancer. Moreover, we know, based on multiple studies, that at least looking at certain classes of p53 mutation are associated with worse outcomes. These are things that we know. And yet, creating prospective trials based on p53 status is a hurdle that we cannot seem to surmount. So, okay, so that's where we're at right now. However, there are other common genetic alterations in head and neck tumors, and one of the things that we have been looking at in our laboratory are those genetic alterations. So this is data, and a selection of data that we published, that's 2022, but actually it came out in early 2023. We looked at a group, or several groups, I should say, of head and neck cancer, clinical head and neck cancer specimens. And we, deep sequencing, all the bells and whistles. And then we looked at association with outcome. Now, these are interesting to me, at least, in that they are surgically resected specimens followed by postoperative radiation to a uniform dose of radiation. These are highly curated patients that got just about the same treatment, because I am interested in that, how tumor mutations affect outcome following treatment. And if you're going to do that, you can't have this big grab bag of different treatments because that is less informative. So anyway, and I said, okay, let's look at the top. They've got to be mutated in at least 10% of specimens because head and neck cancer is not an extraordinarily common tumor. It's not like lung cancer. It's not like breast cancer. So if you have mutations in 3% of specimens, that's going to be hard to be actionable. Doable potentially, but hard. So we looked at the different mutations, and you can see obviously p53 is the first. And you can say, Heath, that's not associated with outcome. That's right on the line. Well, this is a binary mutation versus no mutation. And we've seen that to really become discriminatory for p53 mutation, you have to classify it a bit because p53 mutations range from no effect to dominant negative to some effect to gain of function, or GOF, gain of function, which actually means it acquires functions that it didn't have before. And keep that gain of function concept in your head because there will be a test on it a little bit later in the talk. Anyway, so two mutations came out of this analysis. One is caspase-8, and it's a very interesting story, and it's not my story. It's actually my colleague, Dr. Pickering's story up in Yale, that's his story to tell. But my story is EP300 CREB-BP, or histone acetyltransferases, particularly the P300 CBP superfamily of histone acetyltransferases, because we're not extraordinarily inventive when it comes to our naming conventions in molecular biology. So we found that both in regards to overall survival and at least from a radiation oncologist perspective, freedom from local regional recurrence was far worse in those tumors that had a mutation in either CBP or P300. So interesting enough, but what does that mean? So what are these genes? What do they do? They do a lot. They're a bridge and link DNA-bound transcription factors or activators to the basal transcription machinery. Moreover, they acetylate, as the name suggests, histone acetyltransferases. And actually, probably a better name for these, and a name that is building up steam, are the lysine acetyltransferases, or CATs, K-A-T, because again, we like cool acronyms. But regardless, they acetylate proteins, including histones, resulting in chromatin remodeling, relaxation of chromatin structure to enable transcription. And finally, they acetylate certain transcription factors, which modulates their activities, positive or negative. They do a lot of stuff. So that's the general principle of what they are. So now, let me change tack for just a second. We know that these tumors express or have worse outcomes following radiation and head and neck. And I don't like bad outcomes. I want to try to fix bad outcomes. That's why I'm in translational science. Because if I didn't want that, I would be in translation. So another thing that we were doing concurrently at the lab at the same time is we were doing this in vivo shRNA screen. I don't get into too many details, but just suffice to say, we used a library of shRNAs that had quote unquote, we called it at the time the FDAome, basically a bunch of genes that could be targeted by drugs that were in phase 1 or greater. So targetable stuff, as well as a library composed of basically DNA damage genes from KEGG pathways and a few other places. So anyway, so then we took this library and put it in 5 cell lines, 2 HPV positive, 3 HPV negative. This is an immunodeficient model. We also did an immunocompetent model, which I'm trying to write the manuscript for, but I don't have the time. But this is an immunodeficient model of human xenografts, human cancer xenografts. We put the library in, we put it in the flank, and then we treated it. We treated it either with conventionally fractionated radiation or a plaque, not the combination, or one or the other. And the nice thing about all that is at the end of the day, what you get is a tumor that you can look at the barcodes for the shRNAs that remain. Those shRNAs that are not there, compared to reference, are hypothesized to either affect tumor growth alone, or if you've treated it with something, responds to the something. And then you look and see what you got. So this is what we got, at least for radiation. On the y-axis are basically negative log p-values using an analysis to try to account for the fact that you have like 10 shRNAs per target, and each one works a little bit differently, and you have multiple comparisons and statistics. So I let my biostatistician take care of that. However, on the x-axis, same thing. So what I wanted to know, like a lot of things affect tumor growth, right? We know a lot of things affect tumor growth. We know a lot of things affect tumor growth in vivo. What I wanted to know is what preferentially affected response to radiation. So that's what is in yellow. It's kind of the things that were more associated with the radiation p-value, or actually log p-value, and less associated with the untreated. Now, that was a very wide spectrum because I didn't want to miss anything, right? When you're doing a screen, I tend to be a little bit broader in my inclusion criteria, so I don't want to miss anything. There's a lot of stuff here. But then I took the stuff and I subjected it to a pathway analysis. And the first pathway that came out was actually N-terminal peptidyl lysine acetylation. So that's interesting, right? Because we were just talking about a couple of histone acetylators or lysine acetylators in general. Cool. So then we further looked at the data, because the nice thing is these tumors are genotyped, right? We know the mutational status of all of them. And what we found, and this is the graph to the left, what we found is that if we took the mutants for CBP or P300 tumors, we don't have a lot of them, but if we took the mutants versus the wild type and did a ratio, what we found is that inhibition of 3 targets were preferential for those tumors that had mutations in CBP or P300. Remember, those were the bad actors in our clinical data analysis. All right. Now, one of them is TTK, and actually TTK was kind of interesting. Corey Spears a few years ago published inhibition of TTK as a radiosensitizer in breast cancer, which was a very nice study. But we didn't kind of pursue that. What we did find is that if you inhibited the gene product, or excuse me, if you inhibited the gene, either CBP or P300, in the mutant tumors, you actually had a very nice radiosensitizing, or predicted, I should say, radiosensitizing effect. On the left-hand side of the screen, you see CBP and EP300 in lollipop plots. The lollipops going up are mutations that are observed in clinical tumors. The lollipops down are mutations observed in cell lines. And I just show that to show you there may be a little bit of preferential mutations in the HAT domain, a little bit. I mean, it's not dramatic. It's not profound, but there seems to be some preference for the HAT domain. And again, that'll come up a little bit. But there are other domains in these proteins or in these genes as well. All right, so I'm going to summarize about 2 years worth of pain in one slide. So we then said, okay, we have all this cool data, clinical data associated with worse outcome, in vivo screening data, it could be preferential for these mutant tumors. Let's do a boatload of in vitro studies, because that's what we do in molecular biology. And what we found is that in native wild type cells, similar to what we found in the in vivo screen, if we inhibited either CBP or P300, we saw no real effect on radiation response. No radiosensitization, no effect on BRCA1 foci, no effect on apoptosis. Now, here's where it gets a little complicated. Particularly because there are other tumor types where these two, if you inhibit one, or if one is mutated and you inhibit the other, it's synthetically lethal. That's not what we found here. What we found is that if you had baseline CBP mutation and you inhibited CBP, what you found with shRNA, what you found was an increase in radiosensitization, an increase in apoptosis, and a reduction in BRCA1 foci following radiation, which is a marker of, well, generally DNA damage, maybe a little bit more towards HR, but generally DNA damage repair, I should say. However, if you inhibited P300, you did not observe that. Conversely, in the mutant EP300 mutant cell lines, we found the converse. You inhibited CBP, nothing happened. You inhibited P300, increased radiosensitization, increased apoptosis, reduction in BRCA1 foci. And that's that tiny, that eye test on the left-hand side of the screen. So, but again, that was a few years worth of data in one slide. And I want to belabor that for you all because that will put people to sleep. All right. The cooler thing really is that we wanted to validate these studies in in vivo models. So this is UMSC47, which is an HPV positive model. Again, this is immunodeficient, so I apologize to all of my immunology colleagues on the phone or out there. But what we found is that when we inhibited CBP with shRNA, we found a profound radiosensitization. And this is conventionally fractionated radiation, by the way, 2 gray times 8 fractions, which is, again, not a curative dose. But at least in, for example, in one of the 2 shRNAs, 7 of 11 animals following that treatment had no observable tumor at the completion of therapy, 3 of 14 in the other. Moreover, survival was significantly impacted in a positive manner by the combination of radiation and inhibition of CBP. And again, notice I've not been making a great deal of distinction between HPV positive and HPV negative at this point, because at least in the in vitro models and in vivo models to some degree, HPV doesn't seem to be affecting this particular phenomenon. Now, P300 and HPV can certainly interact in a variety of different ways. There's some very nice studies out there, but at least in this context, the radiosensitizing context, we weren't observing a lot of effects of HPV, which is odd for head and neck cancer. So this is an HPV negative model, MSC22A, and we saw even more profound effects. Now, we did see significant effects of the CBP knockdown by itself, but if we combine that with radiation, we saw, again, about half of the animals had no observable tumor following the completion of the experiment. So again, very cool stuff. Of course, human tumors don't have shRNA to any particular thing injected into them. We have to target them with something. Skipped ahead. This is a wild type model. Forget what I just said. Drop what I just said. This is a wild type model. And we performed this in response to reviewers with the valid criticism is, hey, you did these mutant models. You need to show in a wild type. It didn't work. Well, we did. In a wild type, it didn't work. There you go. Negative data. It's good negative data, though, at least in the setting that it supports the hypothesis, right? Because remember, the hypothesis is this only works in a mutant context, right? In a cognate mutant context. And in this wild type context, it did not work. So now, targetability. That's why I put that purple slide in there to remind me that we're going to move on to targetability. All right, so targetability. Is it targetable? Is it clinically targetable? So there is a drug out there, and actually I was just talking to Nabil prior to this talk about, I think a few years ago, it was introduced into a liquid tumor clinical trial. And both Nabil and I were trying to remember which liquid tumor it was, and neither of us could. But regardless, so this drug, ICG-001, which is the one you usually use in vitro, but also PRI-724, it binds to CBP, which is the protein product of PrEP-BP. Now, this is sold, if you go on Select Biochem or whatever, they'll say this is a beta-catenin inhibitor because it affects the interaction with CBP and beta-catenin. However, there's some decent corpus actually of recent data that show that it has a broader inhibitory effect on CBP than just this beta-catenin interaction. And so we tested it. And on the left, this is a compilation of, again, that's more like 3 to 4 months of data, basically showing that surviving fraction in mutants, and I don't know if you can see this arrow, but 22A, 47, HN5 are all mutants. Oh, and it's listed up there. Showed a significant radiosensitization following, this is a SF4. However, that was not observed in 25 SEC152, which is an HPV positive model, and FADU. Again, maybe modest effect here, but certainly not significant. So again, adhering to the same hypothesis that we were just describing. Moreover, this drug has passed through phase 1 and is currently phase 2, but I don't know where that stands right now. Primarily because it only seems to target, it only targets CBP, and that's kind of half the story right now. Now, what are other potential inhibitors of these two, ultimately the two protein products in these genes? Well, there's a few. And there's, we talked about ICG-001, for example. There are TAS1 inhibitors, bromodomain inhibitors, and HAT inhibitors, et cetera. Now, we tested in vitro a couple of different bromodomain inhibitors, and we didn't see much, which was actually disappointing, because those are the ones that we started with. Genentech seems to have some interest in them, so we looked at them. Unfortunately, no effect. However, we then looked at this, the A485, the AbbVie compound, a HAT inhibitor, which is relatively specific for P300 and CBP. Historically, histone acetyltransferases have been hard to target for a variety of reasons that I'm sure medicinal chemists can explain far better than I. But the A485 drugs seem to be a bit more targeted. So we tried it in vitro, because that's what you do. And what we found is that either in CBP or P300 mutant cell lines, inhibition with this agent led to profound radiosensitization, regardless really of background. However, consistent with the hypothesis, if you had both CBP or P300 wild type, you had no effect. So again, consistent with the hypothesis and consistent with the fact that what we may be looking at here is an acetylation event, at least in regards to effects on radiation. Moreover, if you look at what actually happens afterward, what we found is that The drug itself leads to significant, on the left, yes, left. On the left, that's a tunnel assay. In cell lines that were mutant, we saw a profound induction of apoptosis following the combination. That should be A485 plus radiation. Now, the neat thing about this drug is there was an A486, which is an inactive enantiomer, an inactive compound very similar to A485, which was a nice comparator, and it had no risk. So again, consistent with the hypothesis. And on the right, are several Western blots for caspase-3, again showing that the combination of radiation plus A485 in mutants, but not wild types, was associated with increased caspase cleavage. Not shown was PARP cleavage and some other measures of apoptosis, but I hope you get the drift at this point. So we then, now this is more recent data, because we finally got this study done. We then tested this in vivo. So this is UMSC22A. And what we found on the left, and this is just a simple flank experiment, what we found is that conventionally fractionated radiation plus A485 led to a pretty profound radiosensitization event that kind of went away when we stopped the drug, figure. But again, this is all the data points after day like 10 are significantly different from all other groups. Moreover, on the right, if you look at body weight, the combination of radiation plus A485 was no worse, did not really have a worse effect on body weight compared to radiation by itself. And I don't know if that was just, they were all moved around. Maybe the radiated mice were moved around more. I'm not certain, because I'm unfortunately not at the point where I'm doing my in vivo experiments anymore. But regardless, I think that at least this is an indicator that it might be. Now, of course, this is not disputive data that this is a safe agent, but it was nice we had the data to show it. All right. However, A485, from what I can gather, this is a second generation HAT inhibitor. From what I can gather, the pharmacokinetics are not everything that people wanted it to be. So as part of ongoing studies, we're actually looking at other ways to target CBP and P300. And one way are these PROTACs, which is a cool concept. Basically, what you do is you have a targeting molecule to something, your protein of interest, linked to a thing which gets you to a degrader, in this case, E3 ubiquitin ligase. So the PROTAC joins the thing that you're interested in, whatever the thing is, the POI or protein of interest, to the thing that will degrade it, leads to a degradation event, and it's gone. Now, the nice thing is, different than like a kinase inhibitor or different than the HAT inhibitor, is that the protein is completely gone. So if you have a protein that's doing the thing that you're interested in, not necessarily by its activity, but by its presence, it's a scaffold, let's say, you've now gotten rid of it, which could be advantageous. At present, there are 3 PROTACs that have been specifically designed to target CBP and P300, both. And this is, and again, I don't like viability assays after radiation. That said, I have the data, so I show it. These are A485 and 2 other PROTACs following 6th grade. And this is a cell titer globe assay. It's good data. It's not perfect data, but it's good data. And what we found is that at least on this assay, these 2 PROTACs have at least some effect on viability following radiation. And in the range of A485. Again, in this assay, a little bit worse, a little bit better, but in the range of A485, which I found interesting. Now, the PROTACs have their own issues. There are thoughts, how do we make them orally bioavailable? Is the penetrance in vitro going to be the same as the penetrance in vivo, et cetera? But it's a very interesting concept. And I think that if we can figure out those issues, it will open up significant vistas towards targeting tumors. But that's a work in progress. Hopefully the grant gets funded. All right. So I've been talking about head and neck cancer, and that's largely what I do these days, other than administer things. But let's talk about other tumor types as well. So this is just data from the TCGA looking at structural variants of CBP or P300. And what you see is there's actually quite a few. So the very first one are liquid tumors, particularly B-cell tumors. Now, I don't study that, and I know, or I believe in my heart, that that mechanism is very distinct from what I'm studying. They're very different tumors. They have a very different CBP and P300, well, primarily CBP, mutational profile. So I'm not really looking at those. But there are quite a few others here that are in my wheelhouse to a greater or lesser degree, and I've outlined a few. Head and neck cancer, obviously, which I study. Cervical cancer, again, treated with radiation, because I'm focused on radiation and radiosensitizers. Non-small cell lung cancer, again, treated with radiation. Esophagogastric cancer, again, treated with radiation, at least for right now. So I wanted to look at some of these. And I started with lung cancer, because I had some lung lines. And what we found is that similarly, but we used a novel assay that the slides are backwards for. But anyway, so we used a novel assay here. Because previously, remember I taught you about DNA damage repair foci and BRCA1 and all that. That's an indirect measure of DNA damage repair activity. This is a more direct measure. You basically take a GFP reporter substrate, one of two that I've used, although it gets fancier. There are some other constructs that are a little fancier than that, but it basically has two differentially mutated GFP genes and a rare cutting I-cell endonuclease. Long story short, if you have the repair type of interest, you see green, and you can measure it, which is cool. So that's a more direct measure of a DNA damage repair process. And in this case, it was either HR and NGJ. Now, again, for my DNA damage repair colleagues out there, I know that there are many different types, and there are variants in all these different types of DNA damage repair, but let me get away with an HEJ versus HR. All right, so now, we looked at this in lung lines. I didn't want to go back and do all the different foci and whatnot, so we looked at this in lung lines. Another reason, or excuse me, we looked at it in head and neck lines, and then we'll look at it in lung lines. But what we found is that in the head and neck lines, similar to what we saw on the foci, that the drug, the HAT inhibitor, led to a profound repression of HR, similar actually to the positive control that we used. However, there was no real effect on an HEJ. Similarly, or excuse me, contrary-wise, I should say, the wild types had no effect on HR with this drug. So then we looked at lung. And if you can't tell, my slides got a little bit out of order. I'm not completely sure how that happened, but my apologies. Let's go back to lung. So anyway, HR repressed by the drug. Now, let's go back to lung. Similar to what we saw in head and neck or clonogenic assays, on the left, these are all CBP wild type, EP300 wild type. No, a little bit of an effect, but remember, the A486 is actually an inactive compound, right? So this, is this related to something else? Is it related to the vehicle? No, anyway, is it related to something else? I don't know. The inactive compound exhibited very similar radiosensitization. However, in the two mutant lines that we tested, we saw, again, a profound radiosensitization in mutant P300 or CBP. So, adhering to the hypothesis that we saw in head and neck cancer. Moreover, in that same DNA damage repair assay, what we found is that in the mutants, inhibition of HAT activity with A485 led to a profound reduction in HR in the mutants with minimal real effects in the wild types. So again, adhering to that hypothesis, may not solely be a head neck dependent phenomena, but more a mutation dependent phenomena, at least in a similar type of, these are squamous, by the way. So with that data in hand, I wanted to go with the limited data that I had in a few different models, or excuse me, a few different clinical models, or excuse me, a few different clinical subtypes, and see if the clinical data held as well. So these are data, they're not great data, but they are data from the TCGA, mined to the best of my ability for patients treated with radiation. The clinical data for TCGA is kind of hit or miss. Some of the sites are very good, some are less so, but regardless, this is my best estimate of patients selected for having received radiation. And similar to the head and neck data, what we found is that mutation in either of these genes led to a reduction in survival in lung cancer as well as cervical squamous cell carcinoma. Moreover, lung had a little bit better annotation, and I could try to select out those patients that did not receive radiation versus those that did. And what we found is that in those that did not receive radiation, mutation in either CBP or P300 was not associated. So again, indirect data that this process, the mutations in these genes, are affecting response to radiation, or outcome following radiation. They're related to radiation. Indirect data, but provocative, to my mind, at least nonetheless. Now, why? Why is this? Why would inhibiting the activity or the presence of these genes only, quote unquote, work, only radiosensitized in tumors or models that have mutations in these? Well, the easiest explanation would be the following. Mutations lead to less genes, less mRNA, lead to less protein, therefore they're easier to inhibit because there's just less of it. That would be the easiest explanation. However, and this is, again, summarizing a decent amount of data, there is minimal correlation between the mutation and expression in either cell lines or tumors, in either mRNA or protein. We did that. A little bit, but not a lot. Moreover, if you look at the degree of knockdown, we did, there's no real association between the degree of knockdown, i.e., we had some wild-type cell lines that the shRNA knocked it down to almost nothing, no radiosensitization. We had some where in the mutant background where it knocked it down just a little bit, but that was enough. Moreover, we did CRISPR knockout in several wild-type lines, and we did not observe radiosensitization. So it doesn't appear to be a dose-dependent phenomenon. It appears to be truly related to the presence of the mutant gene, protein, etc. Now, if you recall, I said there would be a quiz about p53 mutant gain of function earlier in the talk. So here's the quiz. It's not about p53, but it is about gain of function. So this is really neat data from the Cancer Cell Line Encyclopedia, published back in 2019. And this is just a tiny figure in a very large paper. But what they found is that they actually identified several gain of function mutations for a particular histone mark. So for a particular histone mark acetylation, they found that a few of these mutations were actually above wild type. They were above baseline. And remember, they were only looking, if you look at all of these, they looked at histone marks. Now, I don't see, there weren't a number of other proteins, for example, that are acetylated by these genes. They were just looking at specific marks. So anyway, they found a few that were associated with gain of function. Now, in the TCGA, there were only 3 mutations predicted by their criteria that might be gain of function. That said, that criteria was very limited in scope, which is to say you're only looking at a few marks. What could there be in regards to gain of function in other, by mutating other parts of the gene? That wasn't really addressed. But at least as proof of principle, as proof of concept, it's there. So let's get back to what these things do. They're acetylators. They do a lot of stuff, but ultimately they're acetylators and they're scaffolds. So what do? That's what they do. So, and they're very common. So what we did is we went back and said, okay, let's look at these marks. Let's look at these marks at baseline in a number of different cellular contexts to see what's going on. What we found is that with the A485, that HAT inhibitor that we described earlier, we saw a reduction in acetylation of several marks, but only in the mutant context. So HN5, double mutant, excuse me, 22A, single mutant, CBP. We saw a significant reduction in acetylation of several histone marks, which was not observed in the wild type background. And we saw something very similar with shRNA to CBP or that ICG PRI 751 drug, the CBP inhibitor that's in clinical trials. So then we said, okay, cool. Well, are these mutations leading to effects on histone and protein acetylation? So what we did is we took one. We actually took the mutation that's in UMSC22A, cloned it. We put it in a few different contexts. We put it in 293 T cells. And this just shows the CBP. And remember, we're putting in this, which is a truncated form of CBP. So truncated at 1.7. CBP is big, very big. So we put it in, and that's where the truncation is. And you can see here, these are 293 T cells, full length and truncated. There's a little bit of that in the 293 Ts. But when we force express this mutant, we have a lot more of that relatively and a lot less of the wild type. And then HN31 and Detroit, both are which normally are wild type. We also force expressed this Q1773X mutant. And again, this is just showing the differential aminoblots. So next, we looked at both global acetylysine by doing an IP for acetylysine and then a blot. So basically, we pulled it all down, and then we did a blot work. And this is where, to my mind at least, it kind of starts to get interesting. This mutation led to a profound increase in global acetyllysine within the cell. In 293Ts, we play with them because they're easy to put things in, also in the 2 wild-type cell lines. So again, we saw that for global acetylation. We also saw it for self-acetylation. CBP can actually acetylate itself, and that affects its function and activity. And what we found is a profound increase when we pulled down acetylysine in the blood for CBP. We saw a profound increase with the force expression of this mutant, which is our kind of candidate potential gain of function mutation. Moreover, we did the same thing, but started looking at histone marks. And we saw similar, although not quite as profound data, that if we force express this mutated CBP version in either 293Ts, HN31s, these are wild-type head necklines, we found a similar increase in acetylation of these marks. And then over here, it's just densitometry of this data. Functionally, again, if you put it in, the acetylation of these marks goes up relatively substantially, perhaps not as profoundly as the data I just showed you, but substantially. Now, again, here's where it gets a little interesting too. If you take this mutation and you plunk it into a wild-type background, you can see a modest but significant increase in HR, at least by that assay that I just described to you, that GFP-based assay, which is interesting because now we're actually, people always tell me it's very easy to inhibit things. Can you make things happen? Well, here we're trying to make things happen. We did. So arguing that at least this mutation may have gain of function effects on DNA damage repair. Moreover, so then the question is why? And here's where it gets fuzzy. This is the final figure of the paper. This is the final figure of the grant, where you're kind of going off into the, I don't know what's going on, but this might be one. So anyway, what we did is we looked for BRCA1 acetylation. There's at least some data out there that suggests that BRCA1 acetylation can actually positively affect its function. There's not a lot of data, but there's some. And I was harkening back to the very original data where we saw kind of a profound reduction in BRCA1 foci when we inhibited CBP and P300 actually following radiation. So what we did is we looked at BRCA1 acetylation in a wild-type background where we force expressed this mutation. What we found was a pretty profound induction of BRCA1 acetylation. This is an IP for BRCA1 as an acetylysine in your blood. And moreover, that was completely abrogated by the treatment with the A4A5 histone acetyltransferase inhibitor. We looked at this in a separate wild-type cell line and saw something very similar. And that if we put in this, again, putative gain of function mutation, we saw an increase in BRCA1 acetylation, which was not present when we put the full length in. And CBP-FL is the full length. This is the mutation. So again, arguing for potential gain of function activity for BRCA1. Now, there's some data out there that suggests that CBP, because remember, it's a transcription, it affects transcription. So there's some data out there that suggests that that affected BRCA1 transcription and ultimate presence in the cell. So we did a lot of work looking at that. And what we found that is, at least in these models, when we did siRNA to CBP and looked for BRCA1, repressing it, we looked for BRCA1 expression, what we found, and this is RT-PCR, what we found is that wasn't the case. That functionally, the blue is inhibition of CBP, and this is BRCA1. So we inhibited CBP, worked great, didn't affect BRCA1 expression. And this in the HN31, the wild type, or the mutant context. And then we treated with A485 in 22As. No, again, statistically significant, but probably not biologically meaningful. And then HN31, where we see no effect, there was some repression, but again, there's no effect in this model. All right, so maybe something's going on with BRCA1. IPs are kind of hard in this setting. We had some real problems with IP with BRCA1, doing the acetylation and whatnot. So a new guy came into my lab and he brought this cool assay, the proximity ligation assay. And ultimately what it does, it allows you to visualize proteins if they're real close to one another, about 40 nanometers or less. And what you do is you have secondary antibodies with oligos. They're ligated in the course of the experiment. Then you basically do functionally a TACMAN analysis to amplify your fluorescent signal, and then you visualize it. So again, the goal here is just to see if two things are pretty close together. And what we found is that in the mutant background, again, the ones that have this phenotype, when we treat with radiation, we see a significant increase in the proximity of CBP and BRCA1, arguing that there's some kind of interaction going. Now, in the HN31s, which are a wild type, we really didn't see much. We actually saw a reduction, in my opinion, in the short-term time point. And then this kind of came a little bit up, but closer, if you look here and here, it's a little bit up, but not as dramatic as the mutants. So still a work in progress, but kind of interesting. And then secondarily, because I did say DNA damage targeting therapy, I was hoping to have a little bit more data in this context when I gave you the title of the talk. But we've also, because we're looking at BRCA, and there's a great deal of interest right now in head and neck cancer in regards to the concept of BRCAness, which is to say, are there genetic alterations other than BRCA mutations that lead to a tumor susceptibility to things that target BRCAness? And the classic example of that is actually PARP inhibition. So we've begun studies now looking at the combination of HAT inhibition and PARP inhibition, similar to what we've been doing with radiation. And what we found is, in my opinion, quite interesting. With the combination of olaparib and A485, this white bar is actually a mutant, it's the MSC22A cell line. We see a pretty profound induction of apoptosis when you combine the two. Compare that to this wild type line, which is, I believe it was HN31. We saw a little bit of an increase, but not nearly the extent we saw in the mutant. Moreover, these are two additional mutants. This is HPV negative, this is HPV positive. What we found is, again, with the combination of A485 and olaparib, particularly strikingly in the 22As, and to some degree, this is additive probably in the HPV positive MSC47 line, we saw an induction of, again, this is the viability assay, which is not my favorite. The clonogenic is kind of the gold standard here, but regardless, provocative data suggesting that this concept may extend beyond simply a radiation response to something that is targetable with an agent, which honestly makes reviewers happy because they don't like to see just radiation papers typically. They want to see something else. Anyway, all right, so what's my proposed model? So my proposed model is in the normal scheme of events, you have self-inhibitory, this is the TAS domain, which is an inhibitory domain of CBP and P300 actually. So in the normal span of events, this helps and auto-regulates the activity of this gene or of this protein. However, in a gain-of-function setting, that auto-inhibitory, that self-regulating function really isn't there. So it's going like gangbusters to acetylate anything and everything. You saw that in the big global acetylation assay that we did. So it's going like gangbusters, acetylating histones, acetylating proteins, acetylating BRCA1. And all of this is inhibited by the A485 agent. And this going like gangbusters acetylation leads to, at least in my opinion, increased DNA damage repair, baseline resistance to radiation, but is sensitive to the HAT inhibition plus radiation. Now, mechanistically, I think BRCA1 is involved. I'm not foolish enough to think that's the only thing, but I think it is involved. So conclusions, mutations in these HATs can drive radioresistance and appear to be targetable. And at least some of these mutations are gain of function. I'm not saying they all are, because I don't think they all are, but at least some of them are. So what we're trying to do in the lab right now is to profile these mutations individually, which is as much effort as it sounds like, to try to figure out which of them are and are not gain of function, because I think that will be important in trying to move this to the clinic. So in conclusion, thank you so much for the opportunity to talk. These are just some pictures of the folks in my department, and I'm happy to take any questions.
Heath, this is a fascinating talk, and I think the implications of this research are just at so many levels, and you can project so many lines from this to different directions. I want to go back to the radiation sensitivity, to the radiation effect. All right, because there are many more things here besides radiation. I think, as you very well alluded to, especially targeting these alterations. How do you foresee testing this in the clinic? Because the question is, I mean, we have so many radiation sensitizers, and what you showed is very compelling. But do you think it would be? How would you test it to differentiate it, for example, from like platinum radiation sensitization? And just interested to hear your thoughts on that.
No, it's an excellent question. It's an excellent question because, I mean, it's interesting. You say we have so many radiosensitizers, but actually we don't have that many, right? I mean, we have platinum-based agents and we have a few others in disease types, but and they're not
perfect.
well, no, it's not that they're not perfect even. They're not actually targeted to be radiation sensitizers. They're cytotoxic chemotherapeutic agents, right? They just happen.
They happen to radiation.
Exactly, right? You put them together and they're super toxic, but they also radiosensitize. So how would we move this into the clinic? Well, I think that there's a few key points here. Firstly, I think you would have to do a selective trial. You could not, and I would not recommend doing this in just all comers. So you'd have to do a genomically targeted trial where you basically take the tumors that have these mutations, and then enroll them and randomize them to either, and this would be mine, and because I would do it just like we did it clinically. Oh, sure, of course. I would do it just like we did clinically, which is to say patients, intermediate risk patients, let's say, that receive surgery, because we're always wondering, and we actually, ECOG has a nice ongoing clinical trial asking that question, should we add something else for those intermediate patients? Well, I could envision a study where you take those intermediate risk patients that have the mutations, and it would have to be a multi-institutional trial, and then you combine either radiation alone or radiation plus HAT inhibition. So that's one way you could take it to the clinic.
Do you think you could potentially foresee, let's say, starting with these first and then follow that with radiation, or do you
No, it would be in combination. It would be in
You think
combination.
it should be combination?
Yeah, because the problem is, I don't know what the drug is going to do on its own. Right? And that's the problem with taking some of these radiosensitizers to clinic, right? Because a lot of them, we chase drugs that have effects on their own. We chase them.
Yeah.
The problem is that I think some of the better radiosensitizers may not have that much effect on them, right? Because if it has a lot of effect on its own, guess what? When you combine it with radiation, the toxicity is going to be worse. And that's particularly problematic in the head and neck, right? So, Mylon, you have a question? Oh, sorry. Mhm. So excellent question. I should have been, and I apologize, I should have prefaced this. So these were all squames. So everything that we're looking at right now are squames. And then the other question is, it's a good question about smoking. Since we were focused, at least in the clinical data, primarily on HPV negative, I mean, most of those patients in that cohort had some smoking history, but that's the problem with the TCGA. I wish it had a better kind of quantitation of smoking. Unfortunately, it's, they give you numbers, but how true they are, I don't know. But I mean, so then the question becomes, I'm sorry, go ahead, ask your second one. And that's, what, and I don't know the answer to that. I mean, it would be very interesting to look at the phylogeny of you've got your pretreatment tumor, right? And you treat, and let's say you have recurrence, and comparing the pretreatment to the recurrence. I tried to do something like that many, many years ago. And it was kind of interesting. The few that I was able to do, the mutational profile was actually less, which was weird. And I kind of dropped it because I didn't know what to make of it. But I mean, it would be a fascinating study, right? Because I don't know. I truly don't know what would happen. My suspicion is that mutations in these genes would be enriched, let's say, after you have a recurrence, for example, right? Because you have the radioresistant clone. It'd be my suspicion. It'd be a wonderful study to do. I've not done it, but it's a great idea. And then the other question is about immunotherapy. So as I was finalizing this talk, I was playing with data because I like to play with data. And I was looking, there's a cohort on CBioPortal is a gateway drug for data analysis, by the way. If you don't know, CBioPortal is a neat little website where they've got all these different clinical studies and associated genetic data and whatnot, and you can play with it for hours if you're a geek like me. Anyway, so I was playing, there's one cohort that was treated with immunotherapy, and what I found fascinating was that unlike if you look at most of the cohorts for mutations in these genes, most of them actually show a worse outcome with CBP and P300. However, if you look at the immunotherapy clinical cohort, and I was so tempted to show this data because I thought it was really cool. You should have. I should have. I know, but well, you know what? Everybody. So I wanted to have at least one talk where we didn't talk, just one. But what we found, or what I saw, is that mutations in these genes were actually associated with better outcomes. So is this some kind of an immunogenic microenvironment? I mean, it could just be an artifact, whatever. But it was cool. So the answer to your question is, the second part of your question for immunotherapy, I don't know, but I think that's going to be a very cool thing. Other data I didn't show, because again, I was running out of time and I didn't know if it fitted with the story. We're actually looking at immunogenic cell death, right? So, and what we saw, at least in the preliminary studies that we put together for the grant, is that the combination of radiation plus HAT inhibitor in the mutant context leads to a significant and specific induction of markers of immunogenic cell death, which would be kind of cool, right? Because then that brings IO in the picture, and then now we're talking, right? But excellent, so Mylan, excellent questions. I wish I had the answers. I don't.
Yeah, that would solve the dilemma, right?
That's so many dilemmas.
For the radiation. There is a question from the audience, and we can take that and maybe go back. This is a question from Ned Waller. He's a leukemia transplant expert. So, Dr. Waller. So he basically is asking that your head neck cell lines that you study have significant differences in radiosensitivity. Can the tumor cell line intrinsic radiosensitivity be explained by the differential functional status in these cell lines?
Oh, I wish. Unfortunately, radio response is not clean by any stretch of the imagination. I think that there is a conglomerate of multiple different genetic alterations in these cells that affect their baseline radiosensitivity. I mean, getting the biggest one out of the way. HPV is the biggest actor in the room, both in vitro and in vivo, although a little less in vivo or in vitro, but anyway, or excuse me, a little less in vivo in the preclinical models that we use, but HPV is a big actor. We actually have data that different P53 types, I published that a long time ago, and others have as well, are affecting intrinsic radiosensitivity. However, I think that all of these things key into processes which are driving intrinsic radiosensitization, right? So I think that the P53 story, the HPV story, this story, other stories that are out there, ultimately converge on particular nodes within DNA damage repair, which is driving the radiosensitivity. It's almost like, it's like Google Maps, right? You put in a thing for Google Maps, it gives you like 5 different routes, right? But it all gets you to the same destination. So I think that's what's happening. But unfortunately, as you know, molecular biology is never as simple as what we would like for it to be. And I'm sure it's far more complex than I'm giving it credit for, but that's my general way of thinking about it.
You had a question.
Mm-hmm. Mm-hmm. We looked at 53BP1 foci, and it didn't, the modulation, like it went up following radiation regardless. So it wasn't really affected by modulation of CBP or P300, at least in vitro. And there's some data that mm-hmm. Mm-hmm. Yep. I think the chromatin state could very well be affected, and we're doing actually what we're doing is kind of cool. We're looking at chromatin states at sites where CBP is. That's a more expensive and longer term set of experiments, but that's ongoing, and that's actually part of the ongoing studies. In fact, we have a bunch of cell pellets we're sending to Curtis soon to do some work in that regard. But you're absolutely right. You're absolutely right. I can't give a DNA damage repair talk without actually mentioning DNA, right? So, but you're absolutely right. Excuse me.
I don't see any other questions in the QAs, and I don't see anyone here asking questions. We want to thank again Dr. Skinner for a thought-provoking and excellent talk, and I look forward to your next talk about immunotherapy implications. Or the immune implications of all this, because that should be as fascinating, I'm sure. I'm supposed to basically read you some final comments here. Grand rounds will resume not next week. Next week there will be no grand rounds, but it will be following the week after with Dr. Erica Reeser-Stringer from the University of Alabama talking on current treatment options for patients with HER2-positive metastatic breast cancer. And so finally, we'll close, and thank you very much.
Thank you, everyone. Thank you for your kind attention.